Hi everyone, welcome back to my channel. In this class, we are going to study the DNA replication. So what is DNA replication then? So it is nothing but way the DNA itself will produce its own copies. That is called as the DNA replication. But it has its own steps and process to form the DNA. So before that, we'll learn about the semi-conservative DNA replication. Okay. So what is this semi-conservative DNA replication? So semi-conservative DNA replication is nothing but it is also the DNA replication. So here see, we all know that where DNA is the what? Double-stranded molecule, right? So this parental DNA in the process of replication, it will get what? Divided with the help of enzyme. With these two strands are separated, okay, into different strands. So with this, see, these two are the parental DNA only. So with these two parental DNA, there will be the attachment of the new DNA strand, okay, that is called as the new DNA strand. And now this whole thing will be form or will be acting as the new DNA, okay. So now can you see here where one strand is of the parental DNA and one more strand is the new DNA, okay. That's why it is called as the semi-conservative DNA, because here only half of the parental DNA, only semi of the parental DNA is conserved here, so it is called as the semi-conservative DNA. And now we'll learn how this new strand or how this new strand of DNA is produced. Did you get to know how this new strand of DNA is produced? No. Okay. Now we'll learn how this new strand of DNA is produced and attached with this old DNA and formed as the new DNA. So that is the nothing but there are some process and steps in the process of DNA replication. So in that way, the first step or the first process is the finding the origin of replication. So what is this origin of replication? So think here, if this is the DNA, okay, here the DNA with this strand and this strand, they are the anti-parallel, right? Their polarity will be anti-parallel, okay, and these nucleotides are attached by the what bond? Hydrogen bonds, and also the what? Base pairs. So in base pairs, we know that adenine will always code with the thymine and guanine will always code with the cytosine. The thing what we should notice is here, adenine and thymine will be combined with only 2 bonds, where guanine and cytosine with the 3 bonds. So here in this DNA, a sequence of adenine and thymine will be there, okay. Think A and T. So this is the what sequence where the adenine and thymine are what? They are coded. So this sequence or this place is called as the what? Origin of replication. What it is called? Origin of replication. So why it is called as the origin of replication is because from this point an enzyme called helicase will starts to work on this place or this exact location, where here that enzyme will act on this and it will start to unwind or it will start to break down these bonds, okay. See here the enzyme called, please remember this enzyme, okay, it is very important, helicase, okay, which helps in breaking down these bonds, okay, where it will come and it will recognize the origin of replication space where the only adenine and thymine sequence will be there, because for helicase it is easy to enact or play its role on these bond rather than the guanine and cytosine bond, because it is stronger, right, where guanine and cytosine is stronger than the adenine and thymine. So this helicase will feel, oh, it is, for me it will be difficult to act on the guanine and cytosine, so let me act on the what, adenine and thymine sequence, so that I can break that strand easily. So this helicase enzyme will start, it will find this location, that is the origin of replication. So helicase, what it will do, it will find the origin of replication and it will start to work. It will start to what? Distract, means it starts to break down these bonds and it will get the, it will make this strand to get separated. So when separating, that structure will looks like, see, like this. Okay, this is the exact DNA. Okay, here it is enacting, so that these two strands, this and this, okay, these two strands will get separated, means they will get far from each other. So when they go far from each other, the shape will be like this, and this shape is called as the replication fork. Is this clear? See, one more time I am going to tell you that this is the DNA strand where adenine and thymine sequence will be there, and that place is called as the origin of replication, because the bond will be bit weaker as compared to the guanine and cytosine. So that this helicase will come on this point and it will start to work, or it will start to play its function by breaking down these bonds, and it will, what it will do, it will make sure to distract these two strands from each other. So these two strands will get away from each other and they will look like this, the Y shape, and this is called as the replication fork, because the structure looks like a fork and the replication is taking place there. So now after this, what will happen? So this is the first step with origin of replication. In that, the helicase enzyme will play its role. Okay. So after that, the replication fork form is formed. Okay. When this is formed, see here, this is the what? Polarity, right? We all know that DNA is the antiparallel. So here will be the hydrogen bonds where the nucleotides are attached with these hydrogen bonds and the base pairs. Now what is the case here? With this one DNA strand is away from the another. These two are separated here. So now what we need? So here we will accept the production or the synthesis of new strand of DNA, right? For that also an enzyme called DNA polymerase. So the enzyme called DNA polymerase, please remember this is very important, where this enzyme will help or it will form the new strand of DNA to form the one more copy of DNA. So with this DNA polymerase, this enzyme is very nocturnal. It has its own conditions to play. What it will say? If I need to produce the new strand of DNA, I have two conditions and you guys have to fulfill it. So what are those two conditions? The very first condition put forth by this DNA polymerase enzyme is, it will say, if at all there is only single strand of DNA, then only I will produce the new strand of DNA. So the first condition is accepted because we have the single strand of DNA, right? So they are separate and now they are single. Okay, the first condition is accepted, and the second condition is, it will say I only synthesize or I only produce the new strand of DNA from the polarity like from 5' to 3', but not from 3' to N'. So here this condition is also accepted. So now the DNA polymerase is ready to produce. Okay, I will produce what? The new strand of DNA. So when this DNA polymerase thinks and it starts to move on here to produce new strand of DNA, it will face one more problem and it will think, oh, I need some or any other signal to produce the new strand of DNA. For that, the one more enzyme will come, that is the RNA primase. So this enzyme will tell to this DNA polymerase enzyme, oh DNA polymerase, I am there, I will help you. So how will this RNA primase will help? It will say, you don't worry, I will produce the what? What this RNA primase will say? I will produce the small segment of the RNA strand that is called as the RNA primer, so that it will give you the signal so that you can start to produce the new strand of DNA. So that what this DNA polymerase will do, it will accept the deal. Okay, you just give me the signal, I will come and I will produce the new strand of DNA. So after that, with attaching to this DNA polymerase will come and it will produce the new strand of DNA from the polarity, that is 5' end to the 3' end. Is this clear? Where here the helicase will be enacting here. Okay, it will be here also the same thing, it will be breaking and the same thing will go on. So now here with this one strand polarities, that is from the old one is from 5' to 3', and here it was easy for this DNA polymerase to produce the new strand of DNA from 5' end to 3' end. But here coming to the one more strand, that is here you can see that polarity is from 3' end to 5' end. Okay, but what was the condition of the DNA polymerase? I only produce the new strand of DNA only from 5' end to 3' end. So that now the DNA polymerase cannot produce the 5' end to 3' end here in this DNA strand, because it's already the same thing here. So what it will do now here, it will start to produce again the RNA primase will be there here. RNA primer will be there here. Okay, so now what DNA polymerase will do? Okay, if it goes like this, I just can't go downward, so now it will start to go backward. How? It will produce the DNA new strand from 5 strand to 3 strand in a fragments, in a small units. Again, now here the RNA primer will be formed. Again from here with this link, what this will do? DNA polymerase, again it will produce the DNA strand. So again same thing, this sequence is formed, and here it will be forming the new strand of DNA in the form of fragments or the units. Okay, so like this it will form the new strand of DNA. Is this clear? Is this clear? So here fragments or the units will be formed, hence there will be some of the space or the tuck it will be. Hence it is called as the lagging strand. Okay, it is not continued, so it is called as the lagging strand. But here it is continued perfectly, so it is called as the leading strand. Okay, see these fragments which are formed here, they are called as the Okazaki fragments. Okay, they are called as the Okazaki fragments. Is this clear? So after these, what this DNA polymerase will say, oh, now I have produced or I have synthesized the new strand of DNA. Right? In order to complete it, what it will say? See how it is. It will say to RNA primase, that is ORNA, now I am not in your need. I don't want your help or anything. So please leave me. So what it says? So now your work is completed. You better leave DNA and go off. So that now where all the RNA prime is there, it will get deleted. Okay, it will get deleted. So after deleting, here the space will be there, right? So here that joint space will be, or these DNA fragments will be attached or connected by the enzyme called DNA ligase or ligase. This enzyme will, what it will do, where these fragments are here, it will start to attach them, okay? It will gather it and it will form the continued new strand of DNA. Is this clear? Is this clear? So this is how the replication of DNA is done. So now see here, can you see that where the one parental DNA strand is present, right? And again the two newly synthesized DNA are produced, and now these two acts as the totally new DNAs. So this is called as the DNA replication, and these all are the process steps which are involved in the DNA replication. Okay, is this clear? I think it's clear for you, and in the next video let me come up with a new more videos. Until then, stay tuned and don't forget to subscribe my channel. If you find my videos are useful to you, please try to share it with your friends, and thank you everyone. Take care. Bye bye.